Hi, everyone. Welcome back to Lonza's podcast on the latest trends in pharma manufacturing. Today's topic has it all: novel therapeutic modalities and delivery systems, working from home due to COVID restrictions, fuel injection in cars, and award-winning nanoscience. Stay tuned to learn more about encapsulating mRNA therapeutics. This episode of A View On focuses on the encapsulation process of mRNA using microfluidics. Imagine delivering life-saving instructions to cells like a perfectly written letter sealed in a nano-sized envelope. This is the essence of mRNA encapsulation, a cutting-edge technology that packages therapeutic blueprints into lipid nanoparticles, ensuring they safely reach their destination. These lipid nanoparticles, also called LNPs, are the powerhouse of this process, acting as precise delivery vehicles that make modern mRNA-based therapies possible. At the heart of today's discussion is a device called FDMix, which produces nano-sized LNPs that can encapsulate a therapeutic cargo. It was jointly developed by the high-tech engineering company FDX Fluid Dynamix, the Fraunhofer Institute in Berlin, and Lonza. It has the potential to revolutionize the manufacturing of mRNA-based therapies. This technology was even shortlisted for the 2024 Medicine Makers Innovation Awards, highlighting its innovative potential for pharmaceutical development.
The interesting thing here is the mRNA is not the therapeutic itself, it's just kind of a precursor, and the real therapeutic is constructed by the body, which is quite remarkable and kind of a new technology as a therapeutic.
But how exactly do these mRNA-based therapies work? How are they delivered to patients, and what does the manufacturing process look like? We'll answer all of these questions and more, including how the FDMix platform streamlines mRNA encapsulation. Stay tuned for the next 20 minutes of A View On, brought to you by Lonza. Today, we're exploring the process of encapsulating mRNA therapies into lipid nanoparticles. And for the first time in the history of our podcast, I have 3 speakers joining me. I'm so excited. So let's welcome Christoph Hein, the head of the Ultra and High Precision Technology Division of the Fraunhofer IPK, Bern-hard Bobusch, the CEO of FDX Fluid Dynamix, and Sönke Stocker, the Executive Director of Innovation and Strategic Growth for mRNA at Lonza. Bern-hard, let's start with you. What can you tell us about you and the FDMix project?
Yes, of course. I'm Bernhard. I'm one of the co-founders and CEO of FDX Fluid Dynamix. I'm an engineer by trade, did my PhD in fluid dynamics, and then started FDX together with 2 friends of mine in 2015, working on fuel injection for gas turbines in cars, so completely different field. We started the work on this back in the day, coming from the microfluidic side, scaled them up and used them for fuel injection in the gas turbine in order to enhance the mixing and reduce pollutant emissions. So we developed the Oxygen technology that creates moving jets, moving flow, oscillating flow that we now use for the FDMix part. So we developed the oscillating geometry, or the geometry that creates the oscillation using CFD simulation and scale up, scale down processes and so on.
Thanks, Bernhard. What about you, Christoph? You work at the Fraunhofer IPK. What was your role in the project?
My role within the development is pretty much having the idea. As it is so often, the FDMix platform was created by chance. So more than 10 years ago, I shared my office with one of the co-founders of FDX, with Jens Wintering. And over the time we stayed in contact and also carried out several projects in the field of manufacturing technologies. So I knew quite well about the strengths of the FDX nozzle technology. When the pandemic started, we had then the idea using the OxyJet nozzle technology by FDX for the production of nanoparticles. And that's pretty much the idea behind it. And after some initial tests with handmade plastic mixers that showed the potential of the technology, and that's more or less the start of everything.
So the pandemic was the driver for this process and the product to actually come alive.
Yes, definitely. So it was in April of 2020.
I mean, back in the days, I had very, very frequently phone calls for a year or something like that before we even met in person, Christoph, because it was back in the pandemic days, and it was really interesting.
Sönke, I think now you have the challenge of explaining how Lonza came to this collaboration, right?
Yeah, I think it was in summer 2021 that I met Christoph, and I discovered that we are both working on mRNA topics at that time. I was just new in my role as head of innovation for business unit mRNA, which was just newly created based on the success that we had with the COVID vaccine manufacturing. When we discovered we both work on mRNA topics, and he was working on a new solution for LNP manufacturing, so the mixing of mRNA with certain lipids in order to get to the famous LNP, so the lipid nanoparticles. It raised my interest, and we reconnected later and established a collaboration in order to test this technology and to further develop it. And eventually, Lonza decided to acquire an exclusive license for this technology and to really commercialize it and bring it into the world.
Okay, great. But let's take a step back from the exciting collaboration for a moment and talk about its impact. So it is intended to encapsulate mRNA therapies. We have heard a lot about them in the past 5 years, but Sönke, could you run us through the basics of mRNA?
mRNA is messenger RNA, which is a type of RNA that serves as template for protein synthesis in cells. So in the end, it's an information molecule that you can manufacture that is then put into the body, usually via injection. And in the body, by the use of this lipid nanoparticle, it enters cells and is then translated into a protein which the body manufactures itself and which is then used as a therapeutic. The interesting thing here is the mRNA is not the therapeutic itself, it's just kind of a precursor, and the real therapeutic is constructed by the body, which is quite remarkable and kind of a new technology as a therapeutic.
And what indications can be targeted using mRNA-based therapies?
In terms of therapeutic targets, there are several indications. Prophylactic vaccines, as we saw with the COVID vaccine, the area of influenza, and also malaria, HPV, RSV, or even rabies are typical prophylactic targets for vaccines. We also see emerging in clinical trials now cancer vaccination, mainly against solid tumors, for example, breast, lung, colon, or liver cancer. We see some protein replacement therapies, like, for example, for metabolic diseases. And very exciting also are new advances in gene editing technologies. And this is really remarkable because it is not only transitionary, like other mRNA therapeutics, it also leads to permanent modification of DNA. You can theoretically also cure diseases.
Oh, so you would just need like one injection and be cured?
Exactly.
Oh, that sounds like Star Trek. That's amazing.
It's very innovative, very new, and it's very promising that we will see new therapeutics on the market in like 10 years that are really revolutionizing medicine, I think. Usually, therapeutic cancer vaccines are much more personalized and based on fragments of epitopes that are expressed by cancer cells. So what we see emerging in the clinic now is that somebody types your cancer, identifies, for example, the 15 most common epitopes of this cancer, and we then manufacture mRNAs for these 15 epitopes, which would be personalized and train your immune system to detect and destroy the cancer.
Oh, wow.
So this is not really in production yet, but we see it emerging in early preclinical trials.
Fascinating. Thank you. On this podcast, we have covered all kinds of manufacturing, ranging from small molecules through biologics, also cell and gene therapies. How is mRNA different?
In terms of manufacturing?
Mm-hmm.
Well, mRNA is looked at as a biologic, but it's not really manufactured by cells. So that is a difference in manufacturing. It is manufactured by what we call an in vitro transcription reaction, which is in the end an enzymatically driven copy of DNA fragments into RNA.
Mm-hmm.
We then purify this mRNA and encapsulate it together with 3 to 5 lipids into the so-called lipid nanoparticle, which we then filtrate and can fill into sterile vials for the use in the clinic. So I would say where it mainly differs is that it's an enzymatically derived process and not really cell-derived.
Mm-hmm. What's the size of the nanoparticle that's formed at the end?
The lipid nanoparticles are typically between 60 and 120 nanometers in size.
And can you control how many mRNA molecules are inside?
More or less, it's a statistical question because the formation of the nanoparticles depends on electrostatic interactions between the mRNA and all the lipids. The lipids and the mRNA try to get into a stable status, and that is then the nanoparticle in the end. So when you have a very good controllable mixing of all the ingredients, then you get a very good nanoparticle with a small size distribution and a very good quality, so a very good morphology of the nanoparticle.
So efficient and reliable mixing leads to a uniform and stable set of lipid nanoparticles. And this uniformity in morphology then translates to a better therapeutic. I think this really highlights the purpose and importance of the FDMix platform.
Yeah, completely right. Yep.
So what is the scale the mixing can be performed at this stage?
With the FDMix platform, you can basically start with very, very preclinical batch sizes of a milliliter or less, and then can go to continuous flow of a liter per minute or more. So in order to give a scale to the audience, I mean, if you kind of use a generic concentration of 0.3 grams per liter of mRNA and the corresponding lipid mixture, then a standard dose of a vaccine was between 30 and 100 micrograms. So you can calculate backwards how many doses of vaccine this would be in a minute. So it's quite easily scaled up, and the process is, I mean, the excipients are extremely expensive at that point. So it's a very important process to get right because it's not reversible.
So between 30 and 100,000 vaccine doses per minute? Oh, that's crazy. Now I'm going to ask maybe a bit of a naive question, but bear with me. So I have a background as a synthetic organic chemist, and for me, if I picture mixing, I picture a big round bottom flask and the little steering bar at the bottom of it. But here we talk microfluidics. So how does the mixing actually take place in the FDMix device?
In the FDMix device, due to the oscillator technology that we use, we basically have a flow-through channel. And this channel at one point divides into 3 channels. And we use the so-called Coanda effect to get an oscillatory flow. So we have the main flow attaching to a wall, slightly disturbed to one side, and then the feedback channel pushes it to the other side. So the main jet that is coming into the mixing chamber, the main flow, is oscillating, for example, 2 dimensions up and down a couple of 100 times per second. So it's extremely fast oscillation, and there are no moving parts involved, no electronics and nothing. It's just the flow itself that creates this oscillation and instability. And in this oscillatory flow, which basically is the stirrer that you mentioned, so we generate the stirrer in the mixer, and we inject the ethanol with the lipids. So we have this kind of stirred flask, but in a continuous flow manner. And to give an idea about the size, the 1 liter per minute scale that I just mentioned has an inner diameter of about 1.6 times 1.2 millimeters. So it's very, very small. The whole thing basically can put on a stamp
Wow.
in case of footprint. So
Okay.
the flow path really is small, but the throughput and this rapid mixing allows for very good scale up.
Mm-hmm. Sönke, is such a small footprint something standard for mRNA manufacturing?
Typically, our manufacturing footprint is relatively small. We use kind of benchtop equipment, especially for scale up and for process development. And so it integrates quite well into the setup and is very convenient to use. Yeah.
But it's one of the smallest parts of the whole process.
Exactly.
Just on the previous episode, we spoke with Atul Mohindra about manufacturing monoclonal antibodies, where we were speaking about bioreactors of the size of 25,000 liters. And now we talk about something that is as small as a stamp. Manufacturing in the pharma industry truly is amazing, and the scales are just interesting. Okay, I wonder how long did this entire innovation project last? It's hard for me to picture the challenges of the whole collaboration, especially since it was done during the pandemic where people were scared to leave their houses.
Yeah, it depends on who you ask. We started with our development, as I said, in 2020. From our point of view, we were finished with the development in summer of 2021. And then we met Sönke. Yeah, we learned that we have something like a academic solution, a very early stage development. And we also learned that there was a very long way to go regarding all the trials, regarding manufacturing technology, regarding preparation for GMP manufacturing. And yeah, looking back to that time, we wouldn't be able to take the whole way without a strong partner like Lonza is.
So here you have it, 3 partners coming together amidst the pandemic to create a unique mixing device that produces lipid nanoparticles. With the whole device fitting onto a single stamp, the microfluidics-powered mixing device can produce 100,000 vaccine doses per minute. So we discussed the size of the nanoparticle already and the importance of mixing. Can you change the size of the nanoparticles that are being produced based on the parameters you set for the microfluidics flow in the process?
The main driving factor of the particle size is the formulation itself, so the lipids and all the excipients that are used. If you change the flow rates, you can move that around a little bit. But basically what the FDMix does, it gives you the fastest and most homogeneous mixing that you can get in this short time frame.
The homogeneity is really a big advantage in pharmaceutical manufacturing. So if you have a very homogeneous mixture of small particles, usually means that you lose less material. And as mRNA is very expensive and also the lipids are very expensive, it is a cost factor, but also a quality factor to have a very homogeneous mix of exactly the size of the nanoparticles that you are aiming for.
Now, looking at this project with an innovation mindset, could you also load other molecules into the nanoparticle that is manufactured using the FDMX platform? What are the limitations there?
We can also use DNA, for example, and it's on a regular basis that customers approach us with DNA projects also. And that works very well. We tested it. And it's also within scope of our license that we acquired from FDX and Fraunhofer. DNA and various forms of RNA, like self-amplifying RNAs, smaller RNAs, siRNAs, all types of nucleic acids in the end can be encapsulated.
Could you then push this also to encapsulate proteins or something like that?
Might be possible. I discussed with a couple of people going into the direction of peptides maybe, or even other APIs that are extremely hydrophobic. So maybe there's the possibility to have hydrophobic small molecules in there as well, depending, of course, on the electrostatic interactions and so on.
Would it be pushing it too much to think about whole cells? Out of curiosity.
Might be possible. So since the beginning, we were always asked if the shear forces within our mixer could lead to a destruction of the mRNA. And we did a lot of trials regarding that topic, and we found out that there's no loss of integrity at all. So starting from these results, it might be possible to have cells in LNPs.
That's super exciting. It also sounds like the field has a lot of room to grow.
And there's a lot of progress and a lot of change also in the landscape. For example, 3 years ago, during the COVID times, the limitations of LNP have been discussed heavily. For example, these ionizable lipids are only well tolerated at relatively low doses, and that works very well for vaccines. But for other applications, like, for example, protein replacement or also gene editing, you would need much higher doses. And therefore, there has been a discussion if LNPs will be replaced by polymer-based systems in the future. that was heavily discussed 3 years ago. It's different now. These discussions more or less calmed down, and LNPs are really back and are the method of choice. There are a lot of developers developing different ionizable lipids and LNPs with certain tissue tropisms so that you can really kind of target your distribution to certain organs. And that may be coupled with a real targeting mechanism like a conjugated antibody or something like that. Could be a solution for the future. So it's interesting, but also currently very much changing field.
And what developments would you expect in the short to mid-term? Because this sounds like this may need another decade or so.
Well, we see that it works for the prophylactic vaccines. So here we will see some successes. We also think that for cancer vaccination, they are very usable. In the midterm, the main question for me is the gene editing. Can it be used in ex vivo gene editing, for example? It's perfectly usable.
I mean, as you mentioned, Sönke, back in the day, everybody was jumping onto those polymer nanoparticles. And I've seen some discussions about those hybrid forms. So this will be a lot of interesting research regarding the drug delivery system that those nanoparticles are on top of what is done in the mRNA and cell and gene editing field.
What about you, Christoph? What excites you most for the future of this technology?
Yeah, it's cell and gene therapy. We are asked a lot regarding the usage of our technology in that field currently. A lot of people want to move to LNP-based technologies. And as I said, we have a technology that can be used in the lab very easily on small scales. It can be scaled, it is very robust, and that leads to new collaborations in the academic field for us.
Yeah, I think maybe in the end, those kind of innovations are always only possible as a team in the collaboration. I mean, it started in the collaboration between Fraunhofer and ourselves. And then together with Lonza, we took the next step. So it is, I think, in the collaboration between the different parties, this really is where those sparks come off, and we can get the traction and get everything going as fast and as efficient as possible.
Unlocking innovation through collaboration, that's a perfect message to finish our discussion with. Thank you all for joining and sharing your insights with us today.
Yeah, thank you very much for having us.
Thank you very much.
Thank you.
What a great discussion that was. We learned how FDMix allows for highly controlled, homogeneous mixing without moving parts, making it a powerful tool for pharmaceutical development and manufacturing. The versatility of FDMix also extends to encapsulating various nucleic acids, peptides, and potentially other APIs, presenting future possibilities in drug delivery and therapeutic innovation. Thanks for listening to a view on mRNA encapsulation with our guests Christoph Hein, Bern-hard Bobusch, and Sönke Stocker. Join us next time as we discuss acid reflux and its potential treatments. If you cannot wait, head over to lonza.com/a-view-on to listen to our previous episodes, subscribe to never miss an episode, and access additional materials and interviewee info. Bye for now.